Hello everyone, and a warm welcome to this Eurofins Genomics webinar, and therefore welcome to the DNA universe. This live webinar is recorded, and we will share after the webinar a short summary with you via email, so there's no need to write down anything you see interesting during the webinar. The topic of today's webinar is SARS CoV-2 and genomics. We have twice 5 solutions to push your research and safeguard your business. But before we start with the webinar, I'd like to share our agenda with you. So first, we introduce the speaker of today, so you get a fair understanding who is talking to you. We will follow that by a brief overview about our company, Eurofins Genomics. The first webinar part will cover our program Safer at Work, explaining all options you can consider when reopening your business safely. The second part will focus on human diagnostics and environmental testing. So this is basically the first 5 solutions. The next part holds the next 5 solutions. It's part 3. And it focuses on the R&D part, especially interesting for pharma, biotech, and academic customers. We close the webinar with a live Q&A session. And please feel welcome to ask us everything you want. So as promised, I'm going to start with the speakers. My name is Julian Schlossmacher. I'm the head of marketing and communication at Eurofins Genomics Europe. And again, I'd like to welcome you to this webinar. With me on my call are my colleagues, Natalie Smith. She is our project sales manager. She is based in the UK. She is with us for a year and a half now, and she is very, very experienced in her field. And I'm very happy that Tobias Podpotka is in this call as well. He is our director of R&D for Eurofins Genomics Europe, and he is one of our great experts. He has done a lot of talks on many conferences, and I'm very happy that he is with us on this call. So hello Natalie, hello Tobias. And with this, I would like to hand over to Natalie.
Hello everyone, and welcome to our Eurofins Genomics SARS CoV-2 presentation. I would like to tell you a little bit about who we are as a company and what we can offer to support your coronavirus work from research to application. As a company, we pride our service on being fast, complete, and reliable. We work hard to support you with dedicated service and specialized technical collaboration to help you choose the best technologies for your project. We can provide all that you need from the planning stage to completion. We would like to know more about your work, so we've created a quick poll to find out. Julian will take you through this now.
Perfect, yes, I will just start the poll. So which of the following statements matches your situation best? Is it A, I just started my COVID-19 research, or are you already doing research on COVID-19? Maybe you're already an expert in this topic and work for this for quite some time now. Or last but not least, you've seen that we have been working with Formula One, and you're just interested to learn more about us. Okay, I see a few of you have already started to vote. So 40% of you just started their research. That's very interesting. 17% of you started just a little expert, have already a little expertise in this topic. 4% feel very confident with this topic. And I'm really surprised 40% saw us during Formula One, and that's their main reason for joining us. Thank you very much. Now we have a way better understanding who is in this call and what to expect from that. And I hand over back to Natalie.
You may or may not be familiar with what we do here at Eurofins Genomics, but our name gives a lot of it away. Over the years, we've grown to become a full spectrum provider. We work with Eurofins intercompany specialists around the world to provide you with all of the services you need from one place. In the human diagnostics area, we offer serology in the form of ELISA test kits and real-time PCR for direct pathogen detection. For environmental testing, we offer real-time PCR to identify and screen for the presence of viral particles via swabs on environmental surfaces. We recently completed a Safer at Work service offer, This helps to take the stress out of returning to the workplace, as we provide a comprehensive plan for keeping both your staff and customers safe while in your care. We also offer research and development solutions, including real-time PCR, NGS, and brand new arrays that can be done for preclinical and phases 1, 2, and 3. Next slide, please. Now I'm going to talk to you a little bit about the Safer at Work suite I just mentioned. This will include consulting services, staff COVID testing by PCR, antibody testing, environmental services testing, wastewater, and product testing. The Eurofins Safer at Work programme was created based on customer demand for a fully comprehensive service designed to help you set up advanced risk management protocols to contribute to limiting the impact of COVID-19 on your workplace and beyond. We can help any company of any size. One of our most recent and recognisable customers is Formula One, as you now know by the last poll. So what can we offer, and how do we make your life easier and your choices fewer? Our solutions include the designing and auditing of COVID-19 related sampling and testing protocols, as well as action plans. These include the testing of employees using PCR or serology testing for various antibodies. We detect the presence of the virus within employees on site or in a given community through wastewater testing. We can test for the virus on all types of environmental surfaces in the workplace to detect potential risks early on and to monitor the effectiveness of cleaning protocols. Our programs use big data and AI technologies, as well as specific algorithms that are applied in real time to help you make the right decisions. Next slide, please. We offer human diagnostic kits for the detection of SARS CoV-2 antibodies. These kits detect IgA, IgG, and IgM antibodies in human serum. We provide all the materials for your testing for batches of 96 reactions. You can find our detailed manuals, accompanying documents, and easily order online. The link will be provided after this. We stock ELISA kits that can be shipped at ambient temperature on the same day and ordered online for larger orders. Please email our support team. So we have an email there that we'll also provide after the call so that you can contact us with any questions or for the larger orders. Next slide, please. Eurofins also offers real-time PCR kits for SARS CoV-2 virus detection. These kits can be easily ordered online, where you'll find the accompanying documentation and manuals. The kits can be shipped on dry ice on the same day. Again, for larger quantities, just email us at support@Eurofins. Next slide, please. We offer surfaced assay kits for the detection of COVID-19. This detects COVID-19 on your work surfaces. ViruSeq screen detects the E gene that encodes the viral shell. ViruSeq ident detects the RNA-dependent RD-RP gene. These kits will include the primers and probes for 96 reactions, along with the positive and negative control reagents. You can order these online by following the link below. The online site also provides the manuals to go with these kits. Next slide, please. What can we do to support your R&D genomic solutions for SARS CoV-2 is probably what you're asking. So here we've got some slides showing that we can identify genetic susceptibility towards infections, assess the changes in host gene expression induced by viral infection, identify drug modes of action for development and repurposing, identify viral variants, and track the infection status of your study cohort. We offer primer and probe sequences for the SARS-CoV-2 virus as recommended by the CDC and the WHO. We also offer ready-to-use control plasmids for the virus. Next slide, please. If you're interested in pathway analysis, then we have lots of options. Microarrays can be used to evaluate transcriptional changes in pathways to identify drug response of immune-relevant genes. We offer pathway technology to identify genetic susceptibility to infection with specialized genotyping arrays from both Thermo Fisher and Illumina. We can also set up customized genotyping panels. There are options for high throughput and smaller sample numbers. NGS technology is a great option for the sequencing of your samples. Whole genome region sequencing can identify SNPs that indicate and match with certain physiological complications or resilience. And now Tobias will talk to you about array-based genotyping solutions.
Thanks, Natalie. Next slide, please. So we will now present an array solution which could be very helpful for your COVID-19 research. As a brief background, our array facility is located in Aarhus in Denmark and is among the largest facilities in Europe. We have a huge experience and process arrays since over 20 years. Currently, we are preparing several hundred thousands of arrays per year, and the amounts we process are constantly growing. One array product which works very good in our hands are the Asium arrays. Thermo just recently teamed up with several researchers to define which genomic information is important for COVID-19 research and define the content for a SARS CoV-2 human genotyping array. It contains relevant genomic regions like GWAS markers to assess susceptibility to COVID-19 and also pharmacogenetic content for drug discovery, as well as genes that are known to be important for the immune response. Actually, the array's results can be delivered quite fast, and depending on your research, there might be some advantages over whole genome sequencing. On next slide, some of the features of the SARS CoV-2 array are displayed. It is possible to perform population-independent genome-wide association studies, also covering low-frequency SNPs, and also several disease risk-associated genes like BRCA, etc. are covered. Blood typing is possible since specific blood types have been higher risk displaying severe disease symptoms. In addition, sample fingerprinting to double-check sample integrity can be done. And very important, of course, also genes which are directly associated with the COVID-19 disease, like the ACE2 receptor, along with markers for immune reactions and inflammation. This also includes non-coding regions like regulation of expression, HLA-associated loci, and pharmacologically interesting regions like for drug responses, etc. Actually, I don't want to go into too much details, but in summary, the content makes a lot of sense, and we are happy to announce that we will be among the first service providers worldwide to offer this array. So now on the next slide, we will switch to transcriptomics and expression analysis. There are two major gene expression platforms available, which are arrays, but also next-generation sequencing. For transcriptome analysis via arrays, we recommend the Clarium arrays, with which we have quite good experience, and for next-generation sequencing, we use platforms from Illumina. The aims of transcriptome sequencing are diverse, but for COVID-19 research, it is mainly of interest to study changes in the host expression induced by the virus, but also more specialized to check the impact of new drugs to overcome the disease. Since we have arrays as well as NGS available, I will present a brief summary of the pros and cons of arrays versus NGS, and it's actually up to you to decide what fits optimal to your project. So on the next slide, so in general, arrays contain specific probes. Therefore, they are limited to a certain species and also have predefined content. So if something of big relevance pops up during the project, it is not possible to just briefly include it. However, that's somehow rarely happening, to be honest. RNA sequencing via NGS is more flexible, since any species can be analyzed and is also possible to find novel transcripts, splice variants, and so on. For a huge dynamic range, meaning the ratio between the highest and the lowest expressed transcript, the array is more robust. If you have a transcript which is present 99%, just exaggerating, RNA-seq will deliver also 99% for this reads for this transcript, which leads to a strong dilution of very low expressed transcripts. For those special constellations, an array is more sensitive in most cases. The coverage is of course more flexible with RNA sequencing, but also most low abundance SNPs are covered in the, for example, Clarium D array. Actually, several of the known non-coding RNAs are also on the Clarium D array. I continue on the next slide with the time to result. We have express options available for both technologies, starting with a few days. Sample types, which can be used as input, are also comparable. It is possible to start with low inputs, and also degraded RNA works with both technologies. Besides poly-A selection, also specials like ribosomal depletion or globin depletion is available to increase the dynamic range of RNA sequencing. The data size is definitely smaller for arrays, but however, the complexity of the data analysis depends very much on the project. There are free softwares available for both technologies, and we can also help for the bio-IT if necessary or needed. In summary, we would recommend the arrays if you are screening especially in high throughput and RNA sequencing for the unbiased detection of transcripts. RNA sequencing is recommended if the discovery of new transcripts or splice variants are of interest. In addition, mixtures of species, like for example, virus and host, can be also analyzed together. So please decide for yourself if an array or RNA sequencing fits better to your project. The solution we want to present on the next slide is the sequencing and assembly of complete SARS-CoV-2 genomes. RNA sequencing can be used to identify viral variants of SARS-2 and can be also used to assemble complete virus genomes. This can provide information about variability across populations or geographical regions and can be also used to track virus evolution over time. Recently, a mutation called D614G was found in the spike protein of the virus, and this mutation seems to increase the infectivity significantly. So it's really important to also sequence complete viruses and not only parts of it. And of course, the full-length genome information can be used or is even needed for drug or vaccine development. In summary, we can analyze samples from most sources in any sample number starting from one sample, and also bio-IT services are available. On the next slide, a brief overview on SARS-CoV-2 is shown. Most of it is probably known. The SARS-2 genome with the according open reading frames is shown on the bottom. Most important is that the virus genome consists of RNA and is almost 30 kb in length. It is almost certain that the virus originates from bats, which are known as reservoir for several coronaviruses. But actually, it's still discussed how SARS-2 was transmitted to humans. For SARS-1, an identical virus was found in zebra cats from an animal market, and therefore its transmission route was quite obvious. But unfortunately for SARS-2, it is not clear so far. Pangolins, which is this little animal up there, are also available on animal markets and are suspicious as intermediate host, since viruses closely related to SARS-2 have been found from them, but let's say that the real SARS-CoV-2 virus hasn't been found neither in pangolins or in bats or in any other animal. The closest relative to SARS-2, which has been found so far, has still 4% sequence difference, and considering the relatively slow evolution of coronaviruses, it is estimated that the last common ancestor to SARS-2 was about 40 to 50 years ago. So on the next slide, we want to present an overview how a project could look like. For a project, we require about 100 nanograms of RNA. The RNA can be from any source, so especially swab RNA is usually strongly degraded, but works fine with our protocol. In regards of CT values from qPCR, we recommend to take samples with lower CTs. Depending on the sensitivity of the qPCR, CTs below 25 to 30 should be fine. As application, the service can be used for real-time disease epidemiology, phylogenetic studies, and of course also virus variant detection, for example, to track antigen evolution. We can accept any amount of samples, so if you just want to test our service, it's also possible to do a project with only one sample. Ribodepletion is included in our optimized workflow, and the library preparation is also delivering strand-specific information, if that's interesting for you. The basic package includes 5 million read pairs, and it will be sequenced on a NovaSeq in a 150 base pair paired end mode. For the bio-IT, we do a mapping on the SARS-CoV-2 reference genome and generate a SNP table, which we of course also provide. In addition, we generate and provide a viral consensus sequence. It is recommended to use virus samples with coverage above 90%, but in theory, it's also possible to generate references with lower coverage. On the next slide, we will finally show some data. So we took 16 positive samples and 2 negative controls and performed 150 paired-end sequencing on a NovaSeq. For comparability, all reads were downsampled to exactly 5 million read pairs. And then we could detect virus in 15 of 16 cells. For one qPCR positive samples, we didn't have any NGS hit, but this sample also had a very high CT of over 40, as you can see on the table. Initially, this project was just to check if we can find SARS-2 RNA at all, but we saw that with our workflow, it is possible to do much more with the data. For the positive samples, we had 6 samples which were covered above 99%, and from those we generated consensus sequences and full genome references. We had to work a little bit on the mapping stringency, but managed to adjust the pipeline that the negative controls are not showing any unspecific SARS-2 mapping. So on the next slide, we generated coverage plots for all analyzed samples. In the y-axis, the coverage is shown in a logarithmic scale, and in the x-axis is the SARS-2 genome displayed. As you can see, the highly covered isolates are gap-free, and for the ones which have a low coverage, the reads are mapping randomly on the genome. And that kind of shows that we are not having a bias towards certain regions. Very happy about that. The samples with low coverage also show that the amount of read duplicates is quite low. So otherwise we would have some really massive peaks there. On the next slide, we zoom a little bit into the coverage plots. The samples with the highest coverage is displayed, another sample with lower coverage, just to show this a little bit more in detail. And here it's nicely visible that there is not a bias towards any regions and that we get quite an even coverage over the genome. Of course, with a few dots, but as even as it can get. On the next slide, we prepared a phylogenetic tree with our newly generated full-length genomes. For that, we downloaded different isolates from diverse geographical regions and calculated a phylogenetic tree. And what is visible is that all our isolates cluster together in one branch, and 3 were even identical. So that was not surprising since all patients were from northwestern Germany, so living in the same geographical region. And just looking at this data, it might be even likely that the patients were in the same or closely related infection chain. So if you're interested in transcriptome sequencing to acquire full-length SARS-CoV-2 genomes, please let us know. We're happy to assist. And with this, I would like to hand over to Natalie again.
Thank you, Tobias. Now you have heard about the types of support and technologies Eurofins can offer for your virology work, and we have summarised the highlights here for you. Whether you're developing drugs, repurposing them, working on vaccines, or working on specific studies for SARS-CoV-2, we have the services to support you. Next slide, please. We're here to help you with your work and look forward to hearing about your projects. We pride ourselves on keeping up to date with the latest technologies via current platforms and techniques for progressing and completing your work. We look forward to hearing from you and finding out where we can help. Now we have a final poll to create new content for the next webinar based on your wishes, so you help to create what we talk about in the next one. I'd also like to invite you to ask questions at the end, and we will take the time to answer those, so please stick around. So Julian is going to set up the poll on here, and let's see. Here we go. So the question is, what would you like to see in the next SARS-CoV-2 webinar? So I'm sure all of you right now are getting lots of emails, lots of information. So if there's something you haven't heard, something you're interested in hearing more about, then please let us know. And if it's also something that's not listed here or you want to give specifics, you can do that in the comments. So I can see you're answering right now. So you answered, what would you like to see next in the webinar? So very few of you said nothing else, 7%, 26% more product details, 30% more performance details, and 63 more case studies and success stories. I think case studies really are good because that will help us to present exactly how it was used and the outcomes. So that's definitely good to know. And we will include that in the next webinar. So now, if you have any questions, there's a little comments tab on the side. And what you can do is you can just type in your question, and then we will do our best to answer it, either myself or Tobias. And what we can't get answered, we will come back to you with in the follow-up email, where you will also get a copy of this webinar. You'll have a link to the next one, and then you'll have answers to your questions there too. So if you have any questions, please put them through. And they can be anything. It can be a technical question, it can be something about the kits, or it can be anything you can think of relating to your work with SARS-CoV-2 that we might be able to answer. Read this. Okay, so first question here is, do you have any experience on the detection of COVID-19 in air samples? So that's quite a good question there, airborne. Tobias, do we have anything in the lab currently that you can answer that question?
So basically, you would probably need a very sensitive assay. So I would probably go for the qPCR, and then it's probably best to define how you would get those samples. So would you get them over a filter, or so what will be the material you provide? And then somehow the virus needs to be washed from that filter, and I guess that would be possible if you have kind of an expectation how many copies you would get from there, and also how you would kind of enrich the virus from air. I would say in general, we might have a solution for that.
Okay, thank you, Tobias. Another question was, can we get the presentation as a PDF? What we'll do afterwards is we will send you a link that will have the actual recording, so you'll get everything in the recording, and then all of the information, the contacts, will be sent in PDF. So what we'll follow up with, you'll get the emails, you'll get the links to specific kits, and so you'll have it all detailed. And if, I guess we could what we'll do is we'll put the most pertinent things on the PDF for you so that you have the information there, and then you'll get the recording to answer that question. And then also we had a question about COVID-19 in wastewater samples. So Tobias, can you answer any questions on that, about the process?
Yes, I mean it's again the question, so how the material should look like, because the expectation would also be that in wastewater samples the virus is strongly diluted. So best way would be to isolate RNA from that sample, and then to start with a qPCR in order to look how many copies you would expect. So yeah, would be the best way to go.
Okay. And then also, there's another question here. We've got somebody else that can also answer that question. So I need to go in and I think have Julian unmute on there. There's Sonia, if we can unmute, please. Julian?
Yes, we have it sorted. Thank you for that.
Okay. Great.
Yeah, so let me just maybe answer to the last two questions on the air testing and the wastewater testing. In fact, these two types of tests are available in the Eurofins universe, just not in the Eurofins Genomics universe. That's basically some tests that our colleagues from the environment division have developed. And on the wastewater part, usually you have the sample collection devices that you add to your water system, and then you are supposed to collect the samples over a certain period of time, ideally over 24 hours, and then from there they basically boil it down and can detect if there has been any virus in the system. And that's a very nice system to monitor the site. And in case you find something, if you find some virus load and some trends, then you can start testing specific people or surfaces. Same is true for the indoor air testing. So it's also from the environment division, and it's a service that's just being finalized at the moment. So I think has been launched or is being launched like next week, something like that. And it's also some sort of device where you collect the air in a room, and then again there is PCR test on that. And if we can send across some additional material after that webinar to give you more details on these two services.
Oh, thank you for that. That was a very thorough answer. So we also have a few more questions here. We've got one: can we use the CDC published primer and probe sequences directly, even for commercial use? So that question, I'm not sure, maybe Tobias, if you're able to answer that. And if you want me to repeat the question, please let me know.
Yeah,
I'm not sure.
Yeah,
good question. So, but I'm not really sure on what she wants to use the primers. So, yeah, probably. I mean, it's published and it should be usable.
Yes, yeah, even, I think for commercial use, yeah. Can they use them?
They are allowed for commercial use. It's usually only the dye and quencher where you have to make sure that you have either license-free version or you have the license. You pay the royalties to use it in a commercial kit, but the sequences are okay.
Okay, great, thank you. Next question is: how do you secure inactivation of the virus when you handle it in your laboratory? So Tobias, how do we secure inactivation?
Of course, it's a very good question, and usually you inactivate at first. So that's your first thing you do when you get like a potential positive sample. You inactivate it by lysing the sample. So we have proprietary protocols there, so cannot go into details, but that's the first thing we are doing when a sample arrives in the lab.
Yeah, so the solution is breaking it up. Okay, next question. We would like to use it to manufacture. Okay, so this looks like it's an add-on here. So this was part of the question when it said when it was asked, can we use the CDC published primer and probe sequences directly, even for commercial use? And the follow-up to that was, we'd like to use it in to manufacture PCR kits. So that was a follow-up to that. I don't know whether that gives you any more on it at all, Tobias.
Yeah, I think so. We should get in contact there, and I'm really happy that Sonja is also on the call. I think you can further comment it. As far as I know, we provide primers worldwide, so it would be also possible to help out in this situation. Should be possible, yes.
Okay. Well, thank you. Are there any more? It looks like we've probably gone through the questions for now, unless anybody has any last minute questions they'd like to send in. And what we will do is we'll take your note on case studies and success stories for the next webinar, and we'll make sure that we modify it to include that. So definitely join us next time, because you will be getting new material from us on the next web call. So Julian, I'm not sure if we wanted just to close out and say goodbye. Oh, we have one last question. Lysis buffer is only efficient to do PCR. So is the lysis buffer only efficient to do the PCR, or DNA extraction is necessary beforehand?
So usually we recommend to do an RNA extraction, because this increases also the sensitivity of the assays. In theory, it's also possible to use like direct lysis, but from our experience, RNA extraction increases the sensitivity.
Okay. Well, thank you everybody for joining us, and we will be back on the 21st with the follow-up to this webinar. Thank you.
Thank you very much, everyone. Have a good day. Bye-bye. Stay safe.
Goodbye.